This is the IEEE Computer Society Distinguished Lecturer Webinar Series brought to you by the Distinguished Visitors Program. The Distinguished Visitors Program delivers tools for individuals at all stages of their professional careers through visits to chapters, offering opportunities for individual interactions, and to the membership through webinars by respected professionals. We have distinguished visitors around the world who cover machine learning, cybersecurity, robotics, big data, cloud computing, blockchain, and cryptography, among others. Chapters can request a distinguished visitor on computer.org/distinguished-visitors. The Distinguished Visitors Program is able to pay up to $1,000 for a visit to a chapter, but by working with other nearby chapters to develop a tour, that amount can be increased. So when your chapter is planning its next event, think of the Distinguished Visitors Program. Welcome to our Distinguished Lecturer Webinar Series. My name is Kerry Cosby, and I'm the Chapters Manager here at the IEEE Computer Society. I oversee our more than 500 professional and student chapters around the world and manage our Distinguished Visitors Program. I'd like to take care of a few housekeeping tasks before we get going. You can ask your questions in the Q&A panel. Dr. Rahman will answer as many questions as he can following the presentation. When you're writing your questions, if it relates to a particular slide, please do your best to reference that slide. The webinar is being recorded, and the slides and recording will be made available after the webinar. Today's discussion is on controlling malaria with agent-based models. I'd like to introduce our speaker. Dr. M. Suhail Rahman is a professor of the CSE department at BUET. He had worked as visiting research fellow at King's College, London, UK, from 2008 to 2011, and again as visiting senior research fellow there between 2014 and 2015. He's a senior member of both IEEE and ACM, member of the American Mathematical Society and London Mathematical Society. He's also a peer review associate college member of EPSRC UK. Dr. Rahman has received the Commonwealth Scholarship, the Commonwealth Fellowship, ACU Titular Fellowship, University College London Big Data Institute Visiting Grant, London Mathematical Society Visiting Grant, the Bangladesh Academy of Sciences Gold Medal, and UGC Award. He has led research and development projects funded by both the British Council, UGC World Bank, ICT Division, Government of Bangladesh, and BUET. He has worked on high-dimensional knapsack problems, sequence alignment problems, data structures and string combinatorics, sufficient conditions for Hamiltonicity, machine learning-based predictors in bioinformatics, and meta-heuristic solutions for hard problems. He's the academic editor of PLOS-1 and associate editor of BMC Research Notes. Dr. Rahman has edited special issues as a guest editor in Theoretical Computer Science, Journal of Graph Algorithms and Applications, Journal of Discrete Algorithms, Fundamenta and Informaticae, etc. He's also served as program committee member in several conference series and of international repute. Dr. Rahman regularly writes reviews, Mathematical Review and ACM Computing Review. As well as being a Computer Society Distinguished Visitor, he's an ACM Distinguished Speaker, and very recently he was elected as a Fellow of the Bangladesh Academy of Sciences. Dr. Rahman, thank you for being here, and I'd like to pass the floor to you.
Thank you, Kerry. Thank you so much for your nice introduction. I welcome all to this presentation. As you can see, the title says Controlling Malaria with Agent-Based Models. I'm going to I have divided the presentation in three slightly skewed parts, because first I will just briefly discuss what I am going to do, then why we want to do this, and finally the most part will cover how we want to do this. Okay, so what we are going to do, we want to control malaria, as the title says. And we all probably know, I'm going to show you briefly in a slide as well, it is a vector-borne disease, and a female mosquito is responsible for this disease. But the main responsibility, of course, goes to a parasite. there are different species who actually plays with us in respect to this particular malaria disease. We will be talking about one particular species, which is called Anopheles vagus. We also briefly talk about another species, which is called Anopheles gambiae. For the African continents, the Anopheles gambiae is the most deadly species, whereas for Bangladesh and South Asian region, one of the dominant species is Anopheles vagus. And as for the parasites, the parasite family is called the Plasmodium, and among the parasite families as well, there are deadliest ones, and the most deadliest one is Thalassosporium. So with this, we have an idea about what we are going to do, and now I'm just going to I'm just going to show you the first the cycle of the malaria disease that we probably have learned back in our high school. I'm not going into the details, but you all probably know that there are two stages in some sense, because the disease grows into the mosquito, and at the same and then when a mosquito bites a human, it comes into the human. So this is basically what I'm going to say about this particular cycle. But then I will go to the main motivation why we are interested to control malaria. This is the statistics that has been prepared by the World Health Organization in the latest report, 2020 World Malaria Report. So this is the statistics from 2019, and these are estimated cases. There are 229 million estimated cases worldwide, 87 endemic countries, and estimated 0.4 million deaths so far. And if you consider only Africa, then you will see 94% of the global share are basically from South African continents. 90% of the global share are from South African continents. And if you come back to South Asian regions, there is only 3% of the global share now belongs to this region, and only 9,000 deaths here. But overall, it is still a global problem, and we want to eradicate malaria where we are still lagging behind. Now we want to just to show you the current situation, the world map. Again, this is from the World Malaria Report 2020 by the World Health Organization. The maroon areas are the cases where we still have indigenous cases. The green areas are safe, they're certified malaria-free, and there are other areas which are still not malaria-free, and the goal is, of course, to make it completely free.
Dr. Rahman, I'm sorry to interrupt. It doesn't seem your slides are advancing. Can you advance your slide to slide number 10?
Okay, sorry. Is it visible now?
Go. Yes, it is. Thank you.
Okay. So what I was mentioning, that the malaria is still a problem worldwide, and hence we want to eradicate the malaria, and this is why we want to work with malaria, and this is why we started working on an agent-based model for malaria. Okay, so what we have here is we will talk about an agent-based model, and an agent-based model is which simulates the actions and interactions of autonomous agents, and it presents a view of their effects. It presents a view of their effects on overall system, how they work and so on. So there are three main components. There are three main components: agents, rules, and environment. So agents are autonomous agents, and they work with some specific rules which actually govern how the system will work, and they interact with the environment, and based on these rules the system works out. And at the end of the day, when we make some changes, if the effect of those changes are visible to us, then we can work on those. So these are interactions of agents between coherent groups, and small changes create dramatic impacts and so on. So we want to work on these agents, and based on the rules and other things and the environmental factors, we would like to get an idea of a particular phenomena, and based on that phenomena, we want to provide some recommendations or some suggestions which would probably alleviate the situation and so on. So I would like to give you a very simple example of an agent-based model first, and that is a well-known agent-based model, which is called wolf and sheep model. I have just given a very simple snapshot from a particular well-known model, agent-based tool, which is called NetLogo. You probably are aware of this particular tool. So here what happens is wolves and sheep wander around the landscape. Wolves look for the sheep to prey on. Each step costs the wolf some energy. They must eat sheep to replenish their energy, and they die when they run out of energy, and so on. This is the situation. So each wolf or sheep has a fixed probability of producing for reproduction at each time step. So this allows the population to continue. And this actually produces an interesting population dynamics. And our goal was to check whether this ecosystem works out or whether there are any problems there. This is a very well-known basic model which comes as part of this tools library. And interestingly, this particular model, as it is shown here with these particular set of rules, is unstable. So to make it stable, we need to introduce more rules. And as we can see, the new rules that are related to the grass and so on. So what happens is if we just add that the sheep has to eat grasses and the grasses need to be replenished and so on, then the ecosystem becomes stable, and we can get interesting and useful information from the model. So this is very important to understand that when you are working with an agent-based model, we need to formulate the right sort of rules, and we need to get the right environment ready so that we can capture the phenomena properly and adequately. So this brings me to the question for our case where we want to model malaria, we want to control the malaria, as I was mentioning before. What would be our goal to capture within our model? Okay, so what we want to do is we want to have a population, human population, as well as a population of the mosquitoes, particularly the Anopheles mosquitoes. In our case, as I was mentioning, we will be focusing on a particular species, which is called Anopheles vagus. So we will have an Anopheles vagus population. And because of this population, we will assume that a number of people will get malaria. Okay? So this will be the basic model, the core model, that there will be mosquito population and human population. In our case, I will discuss shortly, we will have the human population will not act as a direct agent. Those will be treated as pseudo agents. But at the end of the day, what we want to see is because of the mosquito population, our human population is getting malaria. Now, the goal is to control malaria. So we need to talk about the interventions. What are the interventions? I will discuss it in a slide. But the main point is when we put an intervention, then we would expect that because of those interventions, there will be some improvement. So there will be there will be a less number of malaria incidences and so on. Again, if we put another intervention, for example, if we provide insecticide-treated nets and so on, mosquito nets and so on, we would expect that because of these mosquito nets, the malaria will be controlled even more. So this is the main idea, and this is our main goal. So with this particular goal in our mind, we want to start working on an agent-based model to capture this particular phenomena. So again, in short, we want to get the life cycle of the mosquito and get the environment ready so that our model can capture the correct scenario status quo, that is, there are human population, mosquito population, mosquitoes are biting human, and they are spreading malaria within the human population. And then we want to put the interventions so we can have an idea about which intervention is providing us the right results from our point of view. Okay, so if we proceed, so we want to study the life cycle of the mosquito and so on. And then after studying the life cycle of the mosquito, we would like to have the system ready for this life cycle. But the generalized life cycle does not always match a particular species, even within the Anopheles mosquito species. Here, for example, we all know that the Anopheles mosquito hatches eggs. Then they have a larval stages, which can be several sub-stages. Then they have a pupal stage, and then probably through being immature adults, they reach the adult stage. So, for example, the first comprehensive model for malaria, agent-based model for malaria, was done for Anopheles gambiae. And this was done by one of my colleagues during his PhD thesis. And in fact, he's the one who introduced me to this particular issue and got me interested to work on this. And then we started working in collaboration. So for Anopheles gambiae, you can see in the figure, this is the life cycle of Anopheles gambiae. But as I was mentioning, we are interested in Anopheles vagus because in the study area, that is the Bandarban of Bangladesh, Anopheles vagus is one of the most dominating species. So if you see the Anopheles vagus life cycle, this is quite different, particularly in the larval stage. There are 4 sub-stages within the larval stage. And also, as we will discuss shortly, the nocturnal activities of Anopheles vagus differs from the nocturnal activities of Anopheles gambiae and other species as well. So we would like to model this particular, which I'm showing you now, this particular life cycle of Anopheles vagus. And as we can see, I will detail on the larval, pupal, and egg hatching and other states. But here, for example, as we can see, we have immature stage, we have mature stage, and within the mature stage, adult stage, we have 3 separate stages. But one is blood meal seeking, one is blood meal digesting, and the last one is gravid. That is when they are ready to hatch eggs. So all these details have to be modeled within the agent-based model with information from the literature and so on. And then we will have the status quo ready within the model, and then we will be able to apply interventions and see how does that work out. Okay, so in the next few slides, I will try to discuss some of the models that are available in the literature, as well as what we have used in our model. For example, we will start with the egg hatching and incubation model, because part of the first step is for a mosquito after laying eggs, it will have to hatch, and then incubation has to go on. So there are temperature-based equations in the literature. We incorporated those in our model as well. We had different versions of our model, but at the end of the day, we found out that there are, in fact, probability-based information, which was collected through another collaboration in Bangladesh with Johns Hopkins Malaria Institute and so on, where 2 of my colleagues worked. And from there, we had some probability-based information. And we later found out this probability-based information provided us a more stable and useful and adequate model. So we have used that in our model. Incorporating temperature is also very, very important, because we have to have an idea about the temperature. And as we will see, most of the models in the literature, most of the equations and models in the literature are temperature-based. So what we have used is we have used a weather profile, and this is the first model to have used a weather profile on a daily basis. The previous comprehensive models, particularly on the Anopheles gambiae, used a temperature, fixed temperature throughout. But one limitation even in our model was that we could not incorporate separate temperature profile, separate weather profile for water and air, which probably could be used, could be extended for future research and so on. The second thing we had to do was to incorporate the rainfall effects. And rainfall effect is very important. It affects the system in 2 contrasting way, in the sense that rainfall affects negatively the larval, pupal, and egg stages, because of rainfall, they got damaged. On the other hand, after the rainfall, they got some more breeding sources. So there is a nice contrasting synergy between these two effects. So we had to incorporate rainfall effect within the system. So if we go back to the stages, I've already discussed egg and hatching. Now the next stage is the larval development. So for the larval development, what we have used the field data. Again, I was mentioning that two of my colleagues worked in a project with Johns Hopkins University, and they collected data. I will come back to this collection as well again in a few slides later. But they, for each larval stages, I mean instar 1, 2, 3, 4, they had some concrete information about probability-based information, and we use those information to incorporate within our model. Again, if we go to the pupal development, the same thing happened in the literature. There are some temperature-based equations, and those have been incorporated in our model as well. But at the same time, we have tried with some probability-based model, which was based on the field data that was collected by two of my colleagues I mentioned before. And it turned out that these probability-based models are performing much better than the temperature equation-based model. So this is for the pupal stage. Now, overall, what we have to do is we have to incorporate a mortality model within the system, how an agent will die, what percent of the agent will die, and so on. To do that, we again search the literature, and within the literature, we find some information, and from our colleagues who went to the field, we got some information and we use those information to get a model for the mortality of the adults. Here, as you can see, there are some equations where some basic ideas have been used. This was done during the PhD of my colleague, Dr. Arifin, and he came up with some of these equations and tested those for Anopheles gambiae and in general for Anopheles species. They had some experts within their group as well who are known as the Anopheles mosquito experts within the world. So we took those equations as the basic equations in our model as well. Similarly, we had to incorporate in our core model, we had to incorporate the mortality concept for immature stages as well. Immature stages means the age, larval, pupal, and other stages. And for those as well, we have used previous knowledge as well as the new knowledge we gather. And based on those, we have some equations which we incorporated within our model. All the details of these equations and the rationale thereof are too technical, and I'm not going to discuss it here. But if you are interested, this can be found in our paper, in fact, in the additional files and so on. Then, once we have settled the mortality model, we need to think about the two aspects we discussed. One is temperature as well as the rainfall. So for the temperature, we have already shown you there are temperature-based equations, and we also have used those equations in our mortality model. But for rainfall, we haven't mentioned anything yet. But I already discussed that rainfall has an interesting synergy because it affects the different stages. And for different stages, when it affects, it basically affects those stages from a mortality point of view. So for rainfall, we again used some previous information, and from using those information, we created a new set of equations which we incorporated in our model. There are some interesting parameters here. I'm not going to discuss this here, but if we have time, we can come back to these slides and discuss all those parameters and detail later. So, with all these basics, we are now ready to implement the model. So if you see the block diagram, what we have is we have agents, we have states, we have state transitions, we have environments, we have aquatic habitat, we have human houses, we have to use the weather data, and so on. So these are the basic components of this model, and this is more, they are interacting with each other. And as I was mentioning in a previous slide, all the agents will follow the rules. They will interact with the environment. That is, they have aquatic habitat, they have human house, they have a mosquito environment, because mosquitoes will be going through their life cycles at different stages, that is at larval, pupal, and then immature adult and adult stages, and so on. And there will be a state transition strategy as well, and using those strategy, the mosquitoes will interact with the environment, and it will continue to live and die, and while doing so, it will bite a human, and when it bites a human, we expect that it will transfer the disease to the human. Please note that I discussed about the mature stages. There are 3 mature stages. One was blood meal seeking, where the adult mosquito will seek for a blood meal. So in this stage, it will go to human habitat. There are blood meal digesting, when it has had a blood meal, and then it wants to rest. And usually it rests within the human habitat, and then it becomes ready to lay eggs. So when it lays eggs, then it starts anew. So the egg will hatch, incubate, then the larva will be created, and pupa will be created, and so on. So this is the overall cycle, as I have mentioned already. And this is the component relation diagram. And then I also just have a snapshot of the class diagram. So I'm not going to go into the details. One detail is that we have not implemented human as agents here, because our simplified conceptual model was that we will assume we want to only focus on the female Anopheles mosquito. In fact, we were somewhat forced to do that because the real data that we had was only for female Anopheles mosquitoes. So because we had to validate our model against the female Anopheles mosquito census, we decided on this one. The human agent is not directly implemented as an agent. I will discuss a bit more when this will be clear. So how do the agent-based model proceed? Each hour, it will produce some reports based on the previous hour, then it will kill some mosquitoes and so on. And then what it will do is it will do the transitions at different stages. So these are the main basic steps. And after an hour, it will repeat this cycle again. So this is the main basic idea of the model. And our granularity was hourly. So in many cases, this becomes very difficult to do things hourly. But in our case, because the number of agents was not that high, we could do this. When there are a lot of agents, I will very briefly try to talk about COVID-19 ABM towards the end. Then we will talk about only human agents. When the number of agents is very, very high, in those cases, working with at so granular level becomes very costly, because the main attraction of an agent-based model is that each agent will interact with the other agent and so on. Okay, so this is that. Now we have to incorporate the mosquito behavior as well. How does a mosquito behave? So there is a spatial factor within the system. And for the spatial factor, we have to consider the spatial location, the resource density. What are the resources? In our case, the resources are the human habitats, that is the homes, because it will go to the home of a human and try to get blood meal, and also the aquatic habitats resources as well, and so on. And the agent's movement will depend on these resources. So spatially, these movements must be captured. And for a mosquito, the movements depends on different sorts of cues and so on. And when it have different resources, it behaves differently within the resource. In the next slide, I will just give a simple flow chart where this will be clear for you. For example, just as an example, in the blood meal seeking stage, it wants to go to a particular home of a human habitat, and it will try to have a blood meal by biting a particular human. And if it does not reach for example, it will move from one place to another. And if it finds that to be a resource, that is, it is the resource that it needs now, it will do what it wants to do within that resource. So If it goes into a home, it will have a logic. If it goes into an aquatic habitat, it will have a different logic, and so on. But those logics are very simple, so you can easily understand what the mosquito is doing. And all these movement of the mosquitoes and the activities of the mosquitoes are dependent on the information that we got about the nocturnal activities of the mosquitoes, which we got from a very old work back in 1971 and so on. So this is a 12-hour schedule of a mosquito in some sense. And this gives us an idea about what percent of activity of a mosquito will be there within a particular time frame and so on. And this was implemented within the model. Another aspect of the spatial aspect is the landscape. We have to have a landscape for the model, because ideally we should have used a GIS model, but unfortunately for Bandarban we didn't have GIS information, so we had to create landscapes. And to create landscapes, we have used a tool, vector land tool, and this looks like this is just a snapshot of vector land tool where we created the landscape. And to create the landscape, we had to use some information from the literature. Again, this literature was again from different sources, and we identified 5 different breeding sources from the literature. Also, we identified the ratio between the household, human household, and these breeding sources. So based on this ratio and based on this information about the breeding sources, we created the landscape. So far, we have discussed the core model as well as some implementation issues, but any ABM must be validated or should be validated if the data is available against the real data. So to do that, we use the data that was available through a project I mentioned before between ICDDRB and the Malaria Institute of Johns Hopkins University. And from there, we got female abundance data for a particular year. And we use that data to validate our model. But note that we were not able to do the validation on a quantitative basis. Rather, what we did was we tried to validate the pattern. I will discuss this in a moment why this is important. So just to give you an idea about the study area, this is the study area. These are the 2 unions of Bandarban. And our colleagues went there and they did many sort of larval collection and sort of scientific other things. This was done mainly by ICDDRB people, and through this exercise they came up with the census information, how many female Anopheles vegas are there in those particular unions and so on. So then when we have this data and when we have implemented our core model, what we did, we tried to validate the model against these data. And as I was mentioning, we actually tried to match the pattern. Why? Because the methodology that was used by the ICDDRB folks, but that they use CDC miniature traps, and depending on how many traps were used and how many houses were engaged, the count could be higher and lower and so on. So this gave us an idea of the pattern rather than the actual quantity of the female abundance. So this is why we actually used the pattern to match our model, and we sort of, we were happy, particularly after consulting with the Anopheles species experts, that the curve I am showing is a sort of a good match with the real case. And this, in fact, we have a number of models. I mentioned that we use temperature-based equations and so on. Between those models, the best model was taken based on the validation based on this curve. So then, once the model is ready, now we want to go to our main goal. As I was mentioning in my previous slides, the main idea is to show or implement the interventions and try to provide recommendations to the policymakers on what to do. For example, what sort of interventions we would like to use in Bandarban, for example. So there are a lot of interventions. I have listed a number here. I may have missed some, but the most popular are ITN, that is insecticide-treated nets. This is slightly different than our usual mosquito nets, which we use in this part of the world. We regularly use it. I do not know whether everyone in the audience are aware of mosquito nets, but ITN also has some insecticides with these mosquito nets. So when a mosquito touches the nets, it gets a touch of insecticide and it usually dies down. IRS is the aerosol spray which we normally use. Larval source management is a concept where the aquatic habitats are given insecticide so that we can control mosquitoes and so on. So in our case, we used ITN, IRS, LSM, and different combinations thereof. So different combinations have been used, and the three main parameters that we have to use in our logic for intervention are coverage, repellency, and mortality. So coverage means how many houses within the landscape will be covered by this particular intervention. In quantitative terms, maybe we are providing 100,000 mosquito nets and so on. Repellency is for a particular mosquito intervention. What is the fraction of mosquitoes who will be repelled by this intervention? And mortality is similar. What is the fraction of mosquitoes who will die through this intervention? So these three parameters, these are not, for example, coverage, we can have an idea about the coverage if we know what sort of funding we have. But for repellency and mortality, these are educated cases. So we had to use different parameter combinations to see what sort of results we can get by applying the interventions. So here I have just shown a logical flowchart for the IRS. The interesting point that we have to understand is that these three main interventions work in three different stages of mosquito. For example, IRS, it works in blood meal digestion stage. So I was mentioning before the adult mosquito goes through different stages. It looks for blood meal. This is the blood meal seeking stage. It then, after having a blood meal, it waits. It rests. This is called the blood meal digestion stage. When the digestion is done, it is ready to lay eggs, then it goes to the gravid stage. So IRS actually works in the blood meal digestion stage. So it has have a blood meal, then it is probably waiting in our home at a corner of the wall and so on, or it has gone out. So this is a very simple logical flowchart that can easily be understood. And in the next slide, I'm going to show you these three interventions in a combined manner. And the concept is the same. As I was mentioning, there are three stages. ITN will work at BMS, blood meal seeking. ISR will work at BMD, blood meal digestion. And LSM will work at the gravid stage when it is going to lay eggs. So the concept is like this: if there are aquatic habitats where we have applied LSM, then the mosquito will not go there to lay eggs. It will try to find some other habitats where it can lay eggs. So this is how the controlling is done. This is the main logic. The same thing when we have insecticide-treated nets in a particular house, then the mosquito will not be able to have the blood meal. And because of that, it will not advance to the next stage. If it has some house where the insecticide-treated net is not there, then it will have the blood meal there. We also implemented a partial versus complete solution where the intervention is not completely implemented, and this is human behavior. We have studied that many people, even after giving the insecticide-treated nets, do not use the nets and so on. So there were some simulations where a certain percent of a household only uses the insecticide-treated nets. On the other hand, coverage was based on the household. So if we say that we are covering 80% of the area, that means 80% of the households are under insecticide-treated net. So this is where we tried to do some realistic simulation and so on. So anyway, then what we did was we had to gauge some parameters and so on. And for, as I was mentioning, we have interventions in isolation, and we had interventions in combination. So for all these interventions, we had three different combinations of three parameters, that is coverage, repellency, and mortality. And then we applied these interventions on the core validated model. The goal, as I mentioned earlier, is just to see what is the status of female abundance after the intervention has been applied. We also applied all three interventions in combination. And for that, we also had a number of parameters, and we tried simulations with all these parameters and so on. So this is the main model and the model after intervention. So now we can just briefly talk about some results. As I was mentioning, the ITNs are applied on BMS stage, that is the blood meal seeking stage. We have applied ITN with all different parameters, as I was showing you in this slide. And now, if we look at the results, as we can see, we have the main curve. The upper curve is the blue curve. It is the baseline female abundance curve. So this is the curve after validation, which nicely follows the pattern. And then we have applied different combinations of ITNs. And as you can see, the different combinations of ITNs have resulted in lower female abundance. So when we have lower female abundance, we hypothesize that the malaria incidence will be lower. So this was our conceptual model, and this was because we had only female abundance data, we modeled the ABM in this way. Similarly, we also have checked the situation of IRS, and as I was mentioning, the IRS works in the blood meal digestion stage. So this is like, again, we can see that here as well, the female abundance is lower than the baseline. We also applied larval source management and so on. So these are all, we see that these are providing us some good results, some better than the others, of course. So we went to the combination. And we had different sorts of combinations, ITN plus LSM, ITN plus IRS, IRS plus LSM, and so on. And again, we can see that the situation is much better when we use combination. And at the end, we used a number of parameter combinations where all three interventions were used. And here, it is easier to show this in a control map. The concept was we here just show the % reduction. And here, each sub-figures actually show you a particular IRS coverage. And from the X and Y axis, we show the ITN and, ITN and LSM coverages and so on. So as you can see that with three combinations, we have a huge reduction in the overall female abundance. So at the end of the day, we found that when we apply all three combinations, and this makes sense as well, we get the best results. But then again, we need to think about the financial situation, whether we have enough funding to provide all three combinations. So for those, we have a lot of simulations ready for the policymakers so they can match the right one, provide with the right funding application and so on. So with this, the results are briefly presented. I'm just going to make some few discussions. For example, there are some limitations. I mean, we have not modeled it for all the species. As I was mentioning, we only modeled it for Anopheles vegas. There were different issues like vector resistance, which we did not incorporate. Also, air temperature, water temperature, these sort of things I mentioned during previous slides and so on. So all these things can be considered. And as well as there are some other interesting concepts. For example, I was mentioning about the different species. The field data suggests that there are 3 dominant vectors. And I'm just showing you the percentage of those vectors. Anopheles vegas was in fact the second most dominant vector. But because we had data for Anopheles vegas, so we decided to model Anopheles vegas. Also, there should be a mixed model where different Anopheles species are working together and so on. So if we just think a little bit outside the total discussion so far we have done, the question is, are these sort of researches helpful? One of my goal for this sort of talk is to entice, particularly the younger audience, to work on agent-based models and so on, because there are many modelers who are comfortable working on mathematical models, but I personally think, humbly, the agent-based model are better to capture these sort of phenomena. And this is not only for agent-based models, but this gives you an idea. This is also from the World Report, Malaria report from the WHO, that these sort of researchers actually helped eradicate the malaria, or it is not eradicated already, but to control the malaria so far. I'm sure these sort of researchers, not only the ABM-based, but other model-based researchers, helped the WHO scientists and others who have been working here, the policymakers, to take decisions on what sort of interventions should be provided in different countries where there are malaria and so on. And this is why I would like to note that we should think about working in those even more, and the policymakers as well would be interested to know about these sort of things and so on. So this is almost at the end of my presentation. I would just like to give an idea, within 3 or 4 slides, what we can do with agent-based models for other diseases, particularly with COVID-19. In all the books, all the reports now, we have a COVID-19 section, so we can think of it as a COVID-19 section in a presentation. We are living with COVID-19, and for COVID-19, we implemented a very basic model. Again, I was mentioning that in COVID-19 model, we had only human agents. Humans are spreading the diseases through the virus and so on. So we implemented a COVID-19 model on Ford County, because Ford County, this is the county of United States, had implemented very good strategies from the very beginning. So we have shown that if we did something wrong, for example, with ABM, this can be done as well. Suppose we have a very good situation with ABM, we can show that if we hadn't taken that intervention, what would have happened otherwise, and so on. So for Ford County, we have these sort of results. We also did the same thing for New York City, and from New York City, we were able to show that if we could focus on a particular group of people for intervention, like the doctors, nurses, and workers, then it gives us better results. So this sort of analysis could be done with agent-based models and so on. Because I'm in a hurry, I think I'm almost at the end of the time frame. So I'm just going to finish with a slide, which is very interesting in its own right. So this actually talks about contact tracing. And there was a term called digital herd immunity coined by a scientist, Indian scientist. And what you probably all know about the herd immunity for COVID-19 and other viral-borne diseases or other infectious diseases. The herd immunity is if 70% or maybe more susceptible population have got the disease or somehow vaccinated, then it is assumed that the disease will die down. Now, this guy proposed an idea of a digital herd immunity. It's a fantastic idea. I took it on. When I heard it, I got amazed, and I started working on it. So the concept is, because we know that for COVID-19, the contact tracing is a very interesting concept, and the contact tracing can be done by smartphones. So the idea is, can we find a threshold that if more than X% of people have smartphones, and we assume that if someone has smartphone, he can be contact traced, then the disease will die down. So if 70% or more people of a country are smartphone holder, and assuming that they would allow contact tracing, then the disease will probably die down. This is the concept of herd immunity. And we show in this particular, through this particular ABM, that the concept of herd immunity is very, very pertinent in current context. So this is almost at the end of my presentation. I would like to acknowledge my collaborators and students who work with me, the patient audience, of course. This is an unprecedented time. Usually, I prefer to interact with the audience, but it is not possible in this current scenario. So, and the other thing is I have used many resources from the internet, so I have to acknowledge those resources as well. So with this, I would like to conclude my presentation. I would be happy to answer any questions if I can understand where to find it up. Yeah, okay. So,
there are a few questions in there. If anybody else has any questions, feel free to add them to the Q&A. Go ahead, Dr. Rahman. Okay, thank you. So email, I can send the email. I mean, I think I should go from the lower end, right, Carrie, to get the first questions first. Okay, fine. How the mosquitoes are modeled, individually or by number? Each mosquito was, in different states, we have modeled the mosquitoes in different ways. Mostly they're based on age group cohorts, because most of the equations were age group specific. So this is why the mosquitoes were modeled mostly age group wise. So hope I answer. Okay. Then, okay, fine. Then the question is, rest of the memory is that some would like to do. Yes, of course. So after the blood meal, they will have. So this continues anew for a number of times until the mosquito dies. So the question was, once the mosquito rests after blood meal, is that same mosquito likely to again feed? Of course. I mean, the mosquito will continue to do that cycle until it dies, and so on. Okay. Then, is there a stepwise refinement approach you would recommend? How many versions of the model did you produce? Okay, fantastic. So I did not discuss this in detail. There is a term that is used by the modelers that is called V&V, validation and verification. So the verification is the question, am I implementing the model right? So the concept that I have, has this been implemented rightly? So usually what they do is they do several versions, the same core model, several versions, and then try to match the output of those different versions. This is called verification. I only focus on validation. Validation handles the question, is this the right model? So are we implementing the right model for the mosquito and so on, or for COVID-19 and so on? So there is a different sort of approaches. There could be a stepwise refinement approaches as well. I think if I understand your question correctly, it might be like this, that I will have an output for the larval stage and try to match the output with the larval stage, then probably after pupal stage and so on. That can be done as well. And there are different approaches, and depending on the data, we can actually have different sorts of approaches. We only had female abundance data, so our work was limited by that. Kerry, I think you can provide the email to all the audience, right? My email address, yes, I can send that in the email later.
Okay. Thank you, thank you. Okay, yes, digital herd immunity. This is not our concept. The credit goes to, I forgot his name, I apologize. I heard a talk by ICTP, and I heard this one. They have, they proposed this concept. I like this concept very much. Okay, the next question. Have you published more about digital? No, we only published an ABM, which sort of validated the concept of digital herd immunity, where we have shown that if, I forgot the percentage, we have actually shown on New York City population that if a certain % of the susceptible population have smartphones, we have had other assumptions, like we only contact traced people 2 days back, because otherwise it becomes computationally very expensive, and we have resource constraints. So this is what we have published, but we did not work on digital herd immunity more other than that, okay? So this is done. Oh, thank you very much. Thank you very much. Our Eid al-Fitr is tomorrow, and I forgot to mention, you probably have been hearing some sounds, chaotic sounds outside because people are celebrating. I apologize for that. Our Eid al-Fitr is tomorrow morning. This is 10 p.m. here now almost. So thank you very much. Thank you for thank you very much. Thank you. What programming language? What programming language is it? Okay, this one was implemented in Java, but different versions. I talked about the verification scheme. The verification scheme in the previous version, previous ABM done by my colleague, Dr. Niaz Arifin, was they had different versions, C++ and also at Java, and then they verify. In our work, we actually had different versions of the same on the same language. Also, what you can do is now there are, I mentioned already that there are different tools like NetLogo. There is a very popular tool on Python, which is called Mesa, I think. You can try that as well. And so this could be a very good start. Then why are mosquitoes in Canada not so mean? You need to ask this someone else. I don't know. I don't know. Then with citizen science, the digital data can be gathered even in malaria, right? I don't really understand what you mean by citizen science, but what sort of, we need to discuss. I didn't fully understand what you mean. Probably we can write another question so that I get the idea. I'm sorry. Thank you so much. Thank you very much as well. Caroline, thank you very much. Vincent, thank you very much. Catherine, thank you. Do you think the land covers, forest, cropland affect the behavior of the vector and human a lot? Do they need to be taken into consideration? Yes, again, what is the goal here? What sort of goal you are pursuing will depend what sort of complexities we want to induce in the ABM. For example, I tell you one thing. Give me 1 second, please, just 1 second. Apologies, I had some malfunction with some of my systems. So the question I was making, for example, a mosquito, when it does not have a blood meal in human habitat, it could go to the kettles, for example, right? Because we did not model that because it would be more complex and probably we would not get much more better insight from what we have got already. So these sort of things, for example, the flight coverage of mosquitoes, how long a mosquito can fly within a certain time frame and so on. We did not model those things in our model, but all these questions are pertinent. For example, if someone could ask, my title is controlling malaria, but I'm not really working on malaria directly. If someone wants to work on malaria directly, probably the first thing will come into mind to model the parasite. COVID-19, for example. Someone might think of modeling the virus, but when we sneeze, there are 10 crores of viruses, 100 millions of viruses. So we cannot do that. So yes, of course, different sorts of behavior could be pertinent to this. And all these things, I think this is better for the experts to answer those things who works on the malaria, Anopheles behavior and so on. But at the end of the day, we had resource constraints as well. And this is why our consideration did not include those. Gathering data with sense. Oh, I see. Of course. The problem is, in our case, Bandarban, this is related to the citizen science, I think. Yes, I think this is Catherine, right? Yeah. So the concept is this is a hill tract area, and we do not have enough mobile coverage there. That is the first thing. Also, they are mostly indigenous people, and as unfortunate as it seems, there are some issues with basic needs there. So we could not manage to have this sort of a data collection program. Maybe it is possible now. We have advanced a lot during the last 5 years, The data was collected in 2012, and we did our ABM in around 2016. Okay, thank you very much. Thank you very much as well. People can learn more about the mosquitoes with smartphones, perhaps. Yes, I think I have touched that. Does the model have more human description beyond the class diagram? I need to talk to my student whether that is ready. If you want that, you can send me an email. I will ask him, and probably I will provide you if that is available. Catherine, thanks. Thank you very much. Thank you. So I think I have covered almost all the questions, Kerry.
Yeah, it looks like you've gotten most of them. And we've pretty much come to the end of our time. Can you hear me?
Yes, I can hear you.
Oh, while you were asking questions, my internet went on and off about 4 times, so I was very worried. Yeah, I think you've covered most of them. You know, I'd like to thank you for giving your presentation today. I'd also like to thank all of the audience for attending. Our next webinar in the Distinguished Lecture Webinar Series will be the applications of WSN in agriculture industry. That'll be by Mohammed Rawedin Muhtkasim. And that'll be on June 11th at 9:30 a.m. Eastern Time. We will also have Distinguished Lecture Series webinar on consensus protocols in blockchain systems by Anand Tripathi on June 15th at 12 p.m. Eastern Time. In May, we have a Standards Activity Board Series webinar on insight of blockchain standards. That'll be on May 25th at 9 a.m. Eastern Time. And from our Build Your Career series, we have Making Conflict Work on June 22nd at 11 a.m. Eastern Time. Registration is now open, and we will be sending you a link to these future events, along with the slides and recording of this webinar. Also, if you'll notice in the related content of your screen, the slides and a survey are available for you. Again, Dr. Rahman, thank you very much for your presentation. It was great.
Thank you, Kerry. I thank the audience for their patience and some nice questions. I would have preferred to have direct interactions, but that is not possible now. So maybe next time we will have a situation where we can give presentation in not only in virtually, but in physical presence as well. Till then, I would like to thank everyone for being present here. Thank you very much, Kerry. Thank you.
Thanks a lot. Good night.